again, be super familiar with your specification points. What is, first of all, the role of tissue fluid? What is it and what purpose does it serve? It's basically a watery fluid that bathes all of our cells and tissues. There isn't a single cell in the body that is found in a dry environment. They're all in a fluid. Remember that water is the universal solvent. All substances are dissolved in water. So all chemical reactions happen in water. So cells need to have a watery environment both inside and out. And they provide cells with a stable environment and a constant exchange of substances. What are the contents then of tissue fluid? It's going to contain nutrients like glucose and amino acids, and also fatty acids, and also oxygen. The tissue fluid is going to be, as we're going to see in a minute, pretty much almost identical to the plasma of blood. Remember that the blood plasma is the non-cell part of blood, minus the proteins that are found in blood, and we're going to see that in a second. It will also, though, contain waste products, because as important as it is for cells to absorb, to take in nutrients and oxygen, they have to also release, get rid of their metabolic waste, such as, for instance, urea and carbon dioxide. These are fine to know as examples. Don't worry about knowing any more waste products. How is it formed? How is it formed? Sorry, just to quickly annotate the little diagram that we had here before. So these here are your actual capillaries. It's pretty obvious, but just to make sure. These, of course, are cells. And this little sort of blue thing here is the tissue fluid. So the tissue fluid, crucially, is outside of the capillaries. Inside the capillaries, it's blood. So this is blood. What are hydrostatic and oncotic pressures? Because we're going to see in a minute how they basically work in a competitive way to each other to finally bring about the formation of the tissue fluid. Hydrostatic pressure is a pressure that is created purely mechanically. So something pushes against a liquid, and the liquid, hydrostatic pressure is a term that people use in physics, not necessarily just in biology. And it just literally describes the fact that a fluid will always exert pressure on the walls of its container. In the specific context here of blood vessels of capillaries, it's the positive pressure, meaning out from in to out, that pushes water. So that will be my hydrostatic pressure. It's the positive pressure that pushes water out that a fluid exerts against the vessels that contain it. It will vary throughout the capillaries, but it remains constant in the tissue fluid. Because capillaries will have a higher hydrostatic pressure on the arterial end, because the blood that came to the capillaries at the arterial end came from higher pressure arteries and arterioles, whereas the capillaries at the venous end are taking in blood at much lower hydrostatic pressure. So that's why we say that it varies between capillaries, basically between arterial and capillaries and venal and capillaries. What is the oncotic pressure, on the other hand? It's the negative pressure, meaning pulling water in. Just do a different color. So the oncotic pressure draws water in, and it has nothing to do with, we call it pressure, but it has nothing to do with hydrostatic pressure. It's caused by the osmotic effects of solutes, in this particular case, proteins. You don't need to know about oncotic pressure in Edexcel and AQA. Make a note of that. But you do need to know about hydrostatic pressure. Although even if you're AQA and Edexcel, there's no harm in knowing about it. Don't feel like you have to memorize it in excruciating detail, but know about it, because just have it in the back of your mind, because it's gonna, so often there's extra content questions between exam boards. Oncotic pressure is constant through capillaries, but it's not constant in the tissue fluid, because it's caused by large plasma proteins that cannot leave the blood. So we said that the endothelial cells that make the lining of the capillaries are leaky, meaning that plasma can leak out of them, whereas cells don't. So what will stay behind in the capillary? Blood cells, white blood cells, red blood cells, and large proteins. So here's a large protein. What are some examples of those large proteins? Antibodies, various other hormones like insulin or like adrenaline, which are proteins. If you're asked for an example, just remember antibodies, that's fine. So how do these forces work together to create the tissue fluid? Hydrostatic pressure is always a net force outwards, and the oncotic pressure is always a net force inwards. And what determines what the overall movement of fluid is going to be, whether it's going to be outwards or inwards, is basically which pressure is stronger, which pressure takes over. So hydrostatic pressure, it's higher in blood plasma than the tissue fluid. And so the direction of force is from the plasma to the tissue fluid. Whereas the oncotic pressure is lower at the blood plasma, otherwise known as more negative. The oncotic pressure here, guys, is basically kind of the same as water potential. But it's so the presence of solutes, such as proteins, effectively lowers the water potential. We call it oncotic in this context only because it has the effect of drawing in fluid, creating volume inside the... If you say water potential, you'll be fine if that helps you to remember it better that way, if you don't want to learn an additional term. So it's lower in the blood plasma, it's more negative because of the pressure, the presence of proteins, and it's higher, less negative in tissue fluid because the proteins haven't passed through the capillaries. And so what is the direction of force? It's tissue fluid to plasma. So how does that whole pressure look like? If you're AQA and Edexcel, you don't need to know the numbers. I don't necessarily think that you need to memorize the numbers anyway, but you need to be able to discuss them, and you need to be able to notice the differences, and you need to be able to evaluate them. So in green here, we have, just use appropriate colors here. So in green here, we have the net hydrostatic pressure. And on this end, so we kind of like separate the process into two halves here. This end is what we call the arterial end of the capillary. Don't think of it as an artery, it's a capillary, but it's the arterial end of the capillary, meaning that it just branched out from arterioles. Arterial end of capillary, just to make it clear. And here we have the venous end of the capillary. So we're going to examine what happens at each end separately, and it's a really good idea to structure your answers like that as well. So at the arterial end of the capillary, we have a net hydrostatic pressure that is outwards by 3.2. It's forcing blood plasma outwards. What do we have in blue here? Is the net oncotic pressure, or you can say water potential gradient, And what can we see that this pressure is doing? It's -2. It's wanting to force liquid back into the capillary. Why is that? Because some fluid has already leaked out, but some proteins have been left behind. And so finally, what is going to be my net movement of liquid? Well, obviously here the hydrostatic pressure is bigger than the oncotic pressure. So my net movement is going to be outwards. Outwards. Outward movement of flow. Let's now look at the venous end. What's the first thing that we notice? That the hydrostatic pressure is much lower. Look, 3.2 compared to 0.5. Why? Because the venous end of the capillaries always take blood that is much lower. So a huge amount of hydrostatic pressure is going to be lost basically when the fluid leaks out. If you think about it, if you have a large amount of fluid inside a blood vessel, it's going to be exerting pressure on the walls. If that fluid leaks out, what's going to happen? The pressure is going to drop. And that's what's happened here. I had an outward flow, a net outward flow. So I've simply got less liquid now in my venous end of my capillaries. So it's got a lower hydrostatic pressure. The oncotic pressure has stayed the same, right? And so, but the difference is that now my net movement is going to be net inflow, or inward movement, you can say. Flow. Okay. Let's have a typical look at a typical question here. It's a multiple choice one this time. Which of the following is true? Hydrostatic pressure remains constant through the capillaries. Hydrostatic pressure varies through the capillaries. Oncotic pressure varies through the capillaries. Oncotic pressure varies throughout the tissue fluid. Pardon me. I'm going to let you guys do that, but before, let's look at our context. Our context is basically all of these possible answers. Our command is that we are explaining which of the following is true. So have a go at finding this. Note that like these all sound roughly correct, don't they? So you're going to have to pay really close attention to the exact phrasing. So, as always with multiple choice questions, right? This tip here. If you don't know the answer exactly, you'll eliminate any obviously incorrect answers and guess from those remaining. That's a pretty standard. I'm sure you've been told that before for multiple choice questions. So, just to solve your curiosity, the correct answer here is B, right? But let's see why. So, hydrostatic pressure remains constant throughout the capillaries. Why is this wrong? Because that's the whole premise of creating the tissue fluid in the first place, right? That hydrostatic pressure is larger, higher at the arterial end than the venous end. So that can't be true. It's not constant. Oncotic pressure varies through the capillaries. It does not, because the fluid that is lost and then the proteins that stay behind, both in the arterial and the venous end. Let's look at the diagram again real quick. The oncotic pressure is the same in both ends of the capillary. The hydrostatic pressure differs. And finally, oncotic pressure varies throughout the tissue fluid. Not really, because the oncotic pressure in the tissue fluid is just going to depend on the amount of liquid and on the solutes in it. So it doesn't vary throughout the tissue fluid.